This is the first of some preps for lecture 16. We're going to continue with gene mapping. And where we left the end of the previous lecture, we had done 3 different crosses and got the map distances between all 3 pairs of genes that we're testing. And we came up with a map, but the map had some kind of inaccuracy in it. We calculated a distance of 6 map units between black and purple, 12.5 between purple and vestigial, and then when we did the larger distance, the between black and vestigial, we got 16.5 map units. And the question was, something is wrong here. One of these numbers is inaccurate compared to the others, is a little bit inaccurate, and we want to find out why. Okay, and so there's another way we can approach mapping 3 genes relative to each other. If we set up the crosses correctly, if we can generate the stocks we need, we can map all 3 genes at once. And it allows us to account for some, not all, but some of what makes the larger of the 3 distances the least accurate. And we'll talk about some other phenomena, interference and linkage maps and so on, just a little bit. And then we'll just extend to mapping genes on the X chromosome. If you do your cross right, it's pretty easy, but either way you should be able to do an accurate map on the X. Okay, so we're going to talk about trihybrid test crosses. So if we make an individual that's heterozygous for the 3 genes, and then cross that individual to a parent that is homozygous recessive for all of them, we can do a quicker and, in the end, more accurate map. And we could end up figuring out the distances between the genes, and we would, if we did that, we would generate the correct distances. Okay, so the data set that we generated should give us something that looks like this, where the numbers are consistent. Okay. Okay, so this is a less labor-intensive one, but there's a little bit more thinking and figuring out involved. And so we're going to talk about double crossovers now. It's been hinted at in previous lectures, and you may have thought of it when I asked you to consider what might be the problem with the distances not adding up. So if you have 2 genes close together on a chromosome, there's a certain chance of there being a crossover between them. But because there's no set locations for crossing over to happen, let me just finish this. The next crossover doesn't necessarily take place over here. It could take place close by. So if you look along the length of a chromosome, if you manage to look at thousands and thousands of eukaryotic chromosomes, you would see that the little chiasmata that showed up along the lengths of the chromosomes would be spaced out all over the place. Sometimes there'd be a whole bunch of them close together and relatively few further on, or in other cases they might look quite evenly spaced. In other cases, there might be a lot that are far apart and then a couple closer together and so on. So it's a kind of a random process to some extent. We will talk about when you're looking at very, very close distances, just a few map units apart, there may be some non-random crossover, which we'll talk about afterwards. But the thing about double crossovers is if 2 crossovers occur between 2 genes, in some cases, depending on how that goes, if we are mapping, for example, the A and the B genes, And the parental combination is the two dominants or the two recessives. And then if we have crossover, one crossover, we get some that are dominant for the A allele and then recessive for the B allele. But what if we have a second crossover between them, and that second crossover takes place also between the A and the B genes? Now what we're going to have is, this is a non-crossover chromosome, AB. Here's the other chromatid, big A, and we've crossed over. Oh, and then we've crossed back, and now we have big B. Now there have been two crossovers on this chromosome, but when we look at it, it doesn't, we're just looking at these two genes. It doesn't look like any crossover has happened. And so we would count that, if that, suppose that's a sperm and it fertilizes an egg and etc. We would think of that as a non-crossover chromosome. And the same down here, this one is non-crossover, a, little b, and here we have the recessive A crossover, and then cross back before we get to the B gene. And so this one is going to look like, recessive alleles. So this is also going to look like a non-crossover chromosome. And we're not able to physically see what's going on in the chromosomes. We only can look at the actual alleles that are passed on, and we infer what went on, what happened. So when we look at the parental types, some of them may have been generated by two crossovers happening, and that means when we're mapping genes, we're counting up how many crossovers have taken place and dividing by the total progeny. But we're missing a bunch of crossovers when the distance is large, because the larger the distance is, the more likely it is that two crossovers can happen in that region. Hopefully that makes sense. So this is an example. It's a nicer drawn than what I had just done. But the idea here is you've got two crossovers that have happened, and a big chunk of this chromosome, chromatid swapped with that one and vice versa. So we have had these two crossover events, and the DNA has been mixed between the parental and paternal, and maternal chromosomes. But because we're stuck only looking at these two genes, we don't know, we can't tell if the chromatid that produces the gamete looks like this one on top here, or looks like this one here. And they mean different things if we're trying to figure out how much crossover has occurred in the area between genes A and B. Okay. Now, we can, when we're looking at a 3-gene cross, we will be able to detect some of the double crossovers. And that is because we have 3 places where we can look at the genotypes. And if the same event occurred, as is shown on the previous slide, but we actually were also monitoring gene D here, we would be able to distinguish this chromosome from and this one, of course, from the ones that are non-crossover, because this one would have all recessive alleles, and that one would have all the dominant alleles. Those are your non-crossover ones. But this one has dominant for A and B, but in between we have the recessive for D, and vice versa. Recessive for A and B here, but with the dominant D allele between. And now we know that 2 crossovers, CO is just the way I abbreviate crossovers. Now we know that 2 crossovers occurred between A and B. And then, because we know that, we can include those crossovers when we calculate the map distances, and we will get a more accurate number. Okay, so mapping 3 genes is more efficient as long as you set things up and do your analysis correctly. It's going to also be more accurate because you can detect and account for double crossover. Not all of them necessarily, but most of them, and that makes your numbers more accurate. So we're going to look at the vestigial black-purple situation again. We're going to write it with correct notation because we know from the previous lecture that these genes are linked. I'll remind you of the phenotypes when we do the lecture. But basically, we're going to make trihybrids that are heterozygous for the vestigial wild type or mutant, for the black gene wild type or mutant, and the purple gene wild type or mutant. And then we're going to do a test cross, right? So here's our trihybrid, and this is our homozygous recessive tester. And when we're just looking at the phenotypes of the progeny, we're only going to use the alleles that came from this one parent, this female parent, because what the tester parent, the male in this case, contributes is just recessive alleles. And so we're really just looking at what happened in this female, where were the crossovers, and how many were there of each. Okay, so when we look at this, we see lots and lots of progeny that are similar to one parent or the other. Okay, so these are our parentals, and I just call them the NCO, non-crossover class. So the largest, we expect to see two categories that are high frequency, and those are the ones where no crossing over occurred. And then we also expect to see two categories with really low frequencies, and these are our two crossover classes. Because having two crossovers is much less frequent than a single crossover, okay? And so these are the genotypes for those. And then there's two intermediate classes, and you use the color coding and the connection to the original parents to determine this is one category and this is the other. These are reciprocal classes because wherever you have a dominant allele in this wild type allele, in this category you have the corresponding mutant on the other, and vice versa. So when you put all these together, you have one mutant and one wild type allele for each of the genes, and that's what we mean when we say reciprocal classes. Excuse me. So this is a single crossover class, and then so is this single crossover. So when we are mapping genes that all three genes are they're all linked, then we expect to see two really big classes, two pairs of sort of in-between classes, and then one really small category, and that's this pair here. And we can determine, simply because there's so few of these, that this is the double crossover category. And that's really important because we're going to use the double crossover class to determine the order of the genes and then organize our data so that we can do our analysis. Part of the trick of doing an analysis like this correctly is just organizing the data, and we'll go through that. Okay, so those are your parentals, your non-crossover ones, your two pairs of single crossover categories grouped to help you with the analysis, and then your double crossover class, which we recognize because it's the least frequent of the categories. Okay, now the very first thing we have to do is determine the gene order. So you should never assume that it's being given to you in the correct order. I often present it alphabetically or reverse alphabetically or something like that. And so then what you would do is you would take the parental classes and compare. So this is going to be the parentals, and then these are going to be your double crossovers. And basically you look at every combination. So purple could be in the middle, vestigial could be in the middle, or black could be in the middle. And then you put in the correct parental genotypes that are shown here. And then you do the double crossover to see what you would get. So if purple is in the middle and vestigial mutant, purple plus, black plus is the parental type, and the other is here, then if we do a double crossover, vestigial and then purple, and then crossover again, black plus. So that's the category we'd get. And this is the other one, vestigial plus, purple plus, and then black. That's the other one, okay? And if we were to look at these ones, then the categories we get, if, for example, if vestigial was in the middle, the double crossover classes would give us all the wild types on one chromosome and all the recessives on the other. And if we look back, that's definitely not what we would get. In fact, that's what we get for the single crossover class. So we know that's not the right order. And then for this one, we would have wild type for the purple, but mutant for the other two. And then mutant for the purple, but wild type for the other two. And again, if we go back, wild type for the purple, but mutant for the other two, that's a single crossover class. And mutant for the purple and wild type for the other two. So the one that agrees is this one. And so that means that, I think it's vestigial, right? Is it? No, it's purple. So purple must be in the middle. Okay. So this is the long-winded way of doing it, and you might be lucky and accidentally, the way this is presented, the very first one that you test actually gives you the correct results. It's not a bad idea to just quickly zip through the other two and just make sure, but there's another way you can do it, and that is to just look at the gene order. So you look at the parental, and then you look at the double crossover class, and you see which one is different. So in this case, the parental mutant for the vestigial, wild type for the black, and wild type for the purple. The one that is different is this one. It's still mutant for vestigial, it's wild type for the black, but the purple is mutant. And so that means that that's the one that is different, and that means that is the one that's in the middle. And the same for this one. This was our original parental type, and when we look at the double crossover, the only one that's different is this one. And so there would have been a crossover in which these two stayed the same, but the allele from the other homologue has been swapped in. And so that must be in the middle, right? I hope that's clear. If it's not, I would suggest just drawing it out, and then that will help. Okay, so then the next thing, so you've determined which gene is in the middle, and now you want to calculate the distance from that middle gene to either flanking gene, and that's the next part. But so it's a very good idea. I strongly recommend you take the time to just rewrite the genes in order, because it makes the analysis of the data much easier. Okay, and so I will stop there, and we will come back with the data all rewritten and ready to analyze.